Welcome to Reactome's Analysis Tools. Reactome offers several analysis tools that you can access through the home page or through clicking the Analysis button at the top right corner of the Pathway browser. If you have qualitative data, Analyse Gene List will allow you to identify pathways that are enriched in your dataset. Here, we are submitting a list of identifiers from the UniProt database, our primary reference identifier. This data might be a list of differentially expressed genes or proteins, organized as a single column, or you can include expression data in multiple columns. When you input your list of genes into Reactome, the analysis tool will perform statistical analysis to identify a list of the pathways that are overrepresented in your dataset, and overlay this information over the Pathway browser. For more information about navigating the Pathway browser, please watch our previous video. If you want to perform quantitative data analysis using your transcriptomic or proteomic data, we also have a suite of advanced tools like Reactome GSA available, which you can reach by clicking on Analyse Gene Expression. Reactome GSA, also known as the Reactome Gene Set Analysis Tool, provides you with multiple analysis methods, such as camera, and allows you to upload your own data. For example, you can upload RNA-seq data in raw or normalized formats. We also have options for proteomic data or microarray data. Reactome GSA enables comparisons to publicly available datasets that other researchers have uploaded to databases, such as the EBI Expression Atlas, EBI Single Cell Expression Atlas, or NCBI Grain Data. After uploading or selecting your dataset, the Reactome GSA tool gives you an opportunity to annotate your data table and select your statistical design. You can also click here to create reports that you can download in the next step. These reports summarize the analysis in Excel, PDF or R script formats. After finishing the analysis, the Reactome GSA tool allows you to see detailed information about genes that are up or down regulated in your dataset, using the Reactome knowledge base as a scaffold. As always, you can reach the Reactome help desk by emailing help@reactome.org if you need further assistance. And remember, there is detailed information about this and other topics in our documentation area. We hope you enjoyed this quick overview of a few of Reactome's pathway analysis tools. Stay tuned for more detailed videos about all of our pathway analysis tools. Thanks for joining us on this quick tour.